Moving on, we are now going to look at the different characteristics of muscles and discuss the types that they have in common. There are 4 main types as shown in the slide that we need to understand. Firstly, we have excitability. This is when the muscles become excited because they have responded to a neurochemical message sent via the central nervous system to tell the certain myofibril to pull on the bone at an articulation to provide movement. A neurochemical message is when the muscles are given energy by the neurons in order to apply a force to cause movement at an articulation. Extensibility is where the muscles stretch beyond their resting limits due to the ballistic movements performed in different sports. An example of this is in javelin, where the elbow joint moves from flexion in the preparation phase to extension to propel the javelin through the air in the execution phase to build up momentum. If we have a lack of extensibility, it could result in our muscles remaining elongated, specifically the tendons and ligaments, which makes them more likely to strain, causing an injury. This is known as spasticity. Contractility is the opposite of extensibility, where the muscle contracts in order to decrease the angle of a joint to make it shorten. Later we are going to talk about antagonistic pairs, but it is where one muscle contracts while the other relaxes to cause movement. So the prime mover is shortening while the antagonist is lengthening, causing some resistance. An example of this is the tibialis anterior on the front of the shin and the gastrocnemius of the calf muscle, which allows the foot to contract at the ankle to cause dorsiflexion at the tibialis anterior. Lastly, we have elasticity. This is when a muscle rebounds to its original length and shape after a contraction once the force is no longer applied. This can happen to any limb which is moved back into place after exercise. An example is in football when the hamstrings pull on the knee to cause flexion of the lower leg in the preparation phase, but once the ball has been kicked, the leg returns to its original position because the hamstrings lengthen. Next, we're going to look at the different types of muscle tissue. In the human body, the structure of these specialized myocytes are not the same, and they can be classified into 3 different types based on their differences in structure, which enable them to carry out a specific function. The 3 types of muscle tissue we need to know about are the skeletal, cardiac, and smooth muscle tissue. Here I have a table showing the similarities and differences of the different muscle tissue. Firstly, in terms of location, there are some clear differences between the muscle tissues. The skeletal muscle tissues are located on the bones of the skeleton. This is why they are important in providing movement in order to pull on the bones to act as levers. The cardiac muscle has a very different function and is located only in the myocardium, as its muscular function helps to pump blood around the body. Also, the smooth muscles are similar to the cardiac muscles, but they are only located in the walls of the visceral organs, such as the blood vessels, digestive tracts, and hollow organs, and in the airways and irises of the eye. In terms of size, the skeletal muscles are very large compared to the cardiac and smooth, which are relatively smaller. This is because the skeletal muscles have to be large in order to extend over a bone in order to pull on it to cause a movement. In terms of appearance, the skeletal muscles are composed of long tubular muscle fibers which consist of sarcomeres. These are repeated to give this tissue the striated look because they slide past each other when the muscle contracts and relaxes. The arrangement of the myosin and actin aids its function to provide movement because it enables these muscles to contract and relax rapidly in order to generate large numbers of ATP without subjecting the fibers to friction. The cardiac muscles are also striated due to the sarcomeres and the structure of the actin and myosin, but instead of forming the long cylindrical fibers like the skeletal muscle tissues, they form a more branched appearance. The smooth muscles, on the other hand, are fusiform. This means they are thicker in the middle and thinner on the ends, and they are not striated like the cardiac muscle and skeletal. This is because they do not contain sarcomeres and have the same formation of actin and myosin. The cardiac and skeletal muscles have large numbers of mitochondria. Specifically though, the cardiac and smooth have the most because it makes them fatigue resistant, so they can constantly keep contracting. They also have intercalated discs which allow neighbouring cardiac muscles to join together and which are densely packed with protein to hold the tissues together to help the heart contract in one coordinated unit. In terms of control, the skeletal muscles are controlled by the somatic nervous system. This means they need to be innervated by nerves in order to contract. This is why they are voluntary and under our conscious control. Whereas the cardiac and smooth muscle are involuntary. This is because they don't need to be innervated by nerves and are automatic in terms of function, because we don't have to tell our heart to beat or tell our blood vessels to vasoconstrict or vasodilate. However, some of the smooth muscle tissues are innervated, but because they are auto-rhythmic like the heart, they only require nerves to speed up or slow down the process. The cardiocytes and the skeletal muscles are the same in terms of the striated appearance, but the cardiomyocytes are shorter and thicker in order to perform their function of ejecting a large stroke volume to spread through the myocardium and being able to form a network of connections with the other cardiocytes in order to facilitate a coordinated contraction. Also, similarly, both the cardiac and skeletal muscles are surrounded by an endomysium, but the neuromuscular junction formed in the skeletal muscles differs from the heart as they are not innervated by the autonomic nerves, like we mentioned. The reason why the cardiac muscle and the smooth muscles are involuntary and contract spontaneously is because they're myogenic, the idea that they originate in the muscle tissue rather than the nerve impulses. In terms of speed of contraction, the skeletal muscles vary in terms of the movement needed at the different muscles. They can be fast in terms of high intensity activities or slow in terms of low intensity, whereas the cardiac and smooth are more rhythmic and controlled. This is in order to contract steadily in order to contain and remain functioning. Next, we are going to look at the skeletal muscles, specifically the internal structures of these, to see how they are structured to perform their highly specialized functions. The skeletal muscles are covered by a cell membrane which is known as the sarcolemma, and it forms a barrier against the environment and allows the electrical impulses to travel in and out of the cell. The skeletal muscles are connected to the bones, which are connected by tendons to allow the muscles to pull on these bones to create movement. Here we have a cross-section of a muscle, a skeletal muscle specifically. Firstly, we have the tendon, which we talked about. It's connects the muscle to the bone. Then we have the epimysium, which is a form of tissue that is mainly for protection. It aims to cover the muscle and reduce the friction between the muscle and the bones to form the outer layer with the tendons. As we move deeper into the muscle, there's also an inner layer of connective tissue which is inside. It divides into sections which are known as a store of nerves, network of capillaries, in order to provide the muscles to function. These further divisions are known as the fascicle, which is a bundle of skeletal fibers in which the perimysium encases, which is the outer tissue. There is a tissue inside these fascicles known as the endomysium, which is also another form of connective tissue that comes into contact specifically with the myofibers inside of the muscle cell. Now we go even deeper into the myofiber, because it is a myofiber because it is long than it is wide, and it means it requires multiple nucleuses so the DNA can remain close to the parts of the cell that need it to allow proteins to be transformed much more efficiently. The nucleuses are positioned in the membrane, which is known as the sarcolemma, and the tubes within the sarcolemma, known as the myofibrils, which are striative fibers that are binded together to hold the myosin and actin filaments. These myofibrils consist of bands which constrict to allow the muscles to contract to produce movement. This happens because the sarcomeres are repeated Z-lines formed from actin and myosin filaments. The A band corresponds to the myosin and the I band is only found in the actin. The myosin is held in place by the stretchy protein titin, which allows the Z-lines to shorten by pulling on them. We're going to come to the sliding filament theory later on in more detail. A neuron has to trigger the myosin to pull along the actin and push both the Z-lines closer together, which allows the muscle to shorten. An example of this is the bicep curl. It contracts by a neuron which is sent from the brain through the components of the muscle to allow the myosin to pull on the Z-lines to bring the actin closer together and produce the force needed to lift the weights. Muscles work in pairs in order to create a coordinated movement. They are known as antagonistic pairs. One muscle is known as the agonist and the other is the antagonist. Here on the slide we have the different movements in the body that are controlled by the different types of muscle roles. So muscles can take the roles of the agonist, the antagonist, the synergist and the fixator. The agonist is the muscle that is directly responsible for the movement, as it provides the force needed to contract concentrically. This means to shorten the muscle. But it isn't always the case that the muscle shortens. In a bicep curl, the biceps brachii is the agonist that contracts concentrically during the upwards phase, but also contracts eccentrically during the downward phase, because they are the prime mover in both cases and responsible for producing a larger force to oppose the movement to lengthen the muscle with control, as shown in the diagram. When one muscle performs a movement, the other opposes it with resistance to return the body to its original position. In this picture, the agonist in a chest pass in netball is the biceps brachii, because it is causing flexion at the elbow joint in order to stabilize the ball, while the triceps are the antagonist and lengthen to apply the resistance to this movement. In the execution phase, the arms move to extension because the triceps are the agonist and is following the joint to lengthen, while the biceps are the antagonist in order to generate the force to execute a powerful pass. The antagonist is the muscle in that pair that opposes the prime mover by creating a resistance, but relaxes to not impede the movement. However, they do not always relax. They do provide some tension to aid the control of the movement, to stop it or slow it down, and play a more important role that stops the agonist from overcontracting by giving control to prevent injury. Another example of this is in badminton, when a player goes for a smash shot. The triceps brachii is the agonist because it allows the player to extend their arm at the elbow in order to reach up to hit the shuttlecock over the net. This is while the antagonist is the biceps brachii, which is lengthening to allow the agonist to pull on against it to make sure that gravity doesn't cause a force that is larger than the upwards force, causing the movement to accelerate downwards too quickly to cause damage to the elbow.